Welcome back, everyone. We're going to resume now with the next three speakers. The next speaker up is Dr. Shane Liddelow. He's an assistant professor in the departments of Neuroscience and Physiology, as well as Ophthalmology. He's a member of the Neuroscience Institute. His work focuses on the most abundant cells in the brain, namely astrocytes, and how they play a potentially critical role in normal aging and a variety of neurodegenerative disorders. Despite being relatively early in his career, Dr. Liddelow has managed to accrue a number of awards, including the Inga Iqbal Award for AD Research at the 2019 Alzheimer's Association International Conference. In 2020, he was honored by the receipt of the Blas Frangioni Young Investigator Research Award. And also in that year, he was named to the list for published papers in the top 1% by citations. His work has made significant contributions to delineating the role of his favorite cells to a variety of disease states. And his talk this afternoon is entitled, "What Do Reactive Astrocytes Really Do?" We look forward to his talk.
Thank you for that kind introduction, Thomas. As has been said by many people already today, thank you also to you and your team for putting together such a really wonderful research day, and also for your ongoing support of our research, both personally and through the ADRC. And I'd also like to thank you for suggesting I was still young. I'm clinging to my youth as much as I can. But what I really want to talk to you about, of course, today, of course, is my favorite cell type, astrocytes, one that we study in my lab in a whole host of different diseases. And what I'll do today is try and bring back a whole accrual of work in different models into how we think they're playing a role in the context of chronic neurodegenerative diseases like Alzheimer's disease. Like Helen, I want to start off by thanking the people who actually did all of the work that I'm going to talk to you about today. I've highlighted them here in orange, as well as other members of the lab here at NYU. And in particular, I really want to highlight two people who have spearheaded the projects I'll talk about, which is an amazing graduate student, Kevin Gutenplan, that I was lucky enough to work with while I was at Stanford. And after the passing of our mentor, he sort of became an adjunct member of our lab here at NYU. And also an amazing postdoc in my lab, Philip Hazel, who's been doing a lot of work that was funded by the Blas Frangioni Foundation Award. And of course, I'd like to, of course, bring up again the amazing support of both Blas Frangioni and the foundation, as well as the NYU ADRC. So I'm going to really jump ahead and essentially say, I'm going to assume that everyone knows what astrocytes are. They are glial cells. They support neuronal health and development. And in the context of diseases, things go wrong. And a lot of the AD GWAS hits, like ApoE, clusterin, as well as other genes, are actually highly expressed in genes that are, or highly present in genes that are expressed by astrocytes. And so what my lab does is really take a lot of these GWAS hits and says, well, if they're in genes that are expressed by astrocytes, how do they change the function of astrocytes from a physiological to a pathological condition? And would an understanding of these functional changes enable us to better develop novel therapies going forward to actually help these patients and treat a wide range of neurodegenerative diseases? So what are these functions that we're really interested in? Well, a beautiful cartoon here that was drawn by Rachel Kim, a graduate student in our lab, shows some of the physiological functions here. And here we're looking at this specific fact that astrocytes provide nutrients and metabolic support to neurons. They're really important for the maturation and formation of connections between neurons, or synaptogenesis. What I'll talk a lot about today is their interactions with the immune system, both the central nervous system resident immune cells, microglia, but also peripheral immune cells like Tregs and Th17 cells. And also they're really important for interactions and maintenance of the blood-brain barrier. And one could imagine that a deficit in any one of these aspects could be really problematic in the context of Alzheimer's disease. We know, for instance, that of course neurons die, and this could occur by not providing them enough nutrients. And of course, we know that prodromally there is a decrease in synapse density. And so we're really interested in whether or not these roles could be due to astrocytes. Now, of course, it may not be as clear-cut as astrocytes to simply losing their physiological function. There's a whole host of other possibilities, and one possibility is maybe they just become a little less efficient at this. So we like to think of this as, maybe they don't stop providing trophic support to neurons, but maybe they just don't provide enough, and so the neurons start to starve. Or maybe another important role that they have, which is the uptake of neurotransmitters, maybe it doesn't occur as efficiently, and then the neurons can become excitotoxic and die, and these things have definitely been shown in the field. Also, in the context of some of the disease pathologies, such as amyloid, we know we're going to have plaque formation, and it's been shown by a number of labs that these astrocytes can become extremely proliferative and corral this scar off to try and stop its permeation into the brain parenchyma, leading to secondary effects. But something that my lab has been really focused on ever since we discovered this a few years ago is the possibility that these astrocytes actually gain a function they didn't have previously under physiological conditions, and that is that they actually produce and then secrete a neurotoxin that is able to kill neurons in these degenerative diseases. So what I want to do is take you on a little bit of a sort of survey of vignettes of studies that we're doing in the lab that have just recently been published because of the support from the Frangioni Foundation and also the ADRC here at NYU. So the last piece of information I want to give you is really some context for these studies I'm going to tell you about as well. Is from a few years ago, we published some work that showed that of the many possible responses that astrocytes could have in the context of diseases, infection, and acute injury, one of them that we defined was really due to an interaction between microglia and astrocytes. And it was really the release of very specific cytokines, namely IL-1 alpha, TNF-alpha, and C1q, that could drive the activation of these astrocytes, which we then were able to modulate both in vitro and in vivo. And really importantly, one of the things that we discovered was not only could we sort of form these cells in a dish or stop them from forming in a mouse brain, we could actually also image them in human postmortem tissue. And we were doing this using a marker, this complement component C3. And more importantly than that, we could see them, we knew how to induce them, but we could actually study their function, which is way more interesting from a scientific standpoint. And we noticed that some of these important functions we sort of mentioned just at the top of the talk were actually lost in these astrocytes. So they didn't produce either structural or functional synapses anymore. They actually decreased their capacity to phagocytose debris. And we now know with some ongoing studies that they're extremely competent phagocytoses of amyloid. So this will be problematic in the context of AD. But also, as I mentioned, we found that these astrocytes were extremely toxic to a whole host of different neurons throughout the brain. And one of the major problems that we had with this original study was, although we had a really good grasp of the mechanisms of induction and functional deficits in rodents, and we knew that at least correlatively they were present in human postmortem tissue, we never knew if the mechanisms that induce this reactive state were the same in human cells. And so just last year, we published some work that was in collaboration with Valentina Fossati's group just across Manhattan here at the New York Stem Cell Foundation. And this was spearheaded by Lily Babar in her lab and also Indigo Rose here in my lab. And what we did was we actually took human iPSC-derived neurons and astrocytes, and using exactly the same methods that we had shown in rodents, we showed we could take the same microglia-derived cytokines to actually induce a reactive phenotype in these astrocytes that was transcriptomically similar to the rodent cells. But most importantly, we could run these functional assays and show that, once again, human cells lost the ability to form new synapses and to actually properly maintain these synapses. These human cells completely lost their ability to phagocytose myelin debris, synapses, and amyloid beta. And importantly, these human cells retained this ability to become neurotoxic and kill neurons. And this was important for us because it gave us the first piece of evidence that this response, this particular substate of reactive astrocytes, was evolutionarily conserved across these species, which makes them an intriguing sort of avenue for ongoing research. So this sort of leads to a sort of a side hustle that we had for a while, which was to study the fact that if these astrocytes can kill neurons, and we know that they are present in the brains of patients with all of these different neurodegenerative diseases, but only in the regions of pathology, why were they not actually killing all of the neurons in the brain? Why was there some apparent susceptibility and regional differences in the neurodegeneration and also the activation. So we're really interested in if astrocytes can kill the neurons, why don't all of the neurons die? And more importantly, if we have the ability to modulate this reactivity and maintain the function, the sort of presence of these neurons, do we actually retain any neurological function that we may then use as a reasoning behind trying to target these cells therapeutically? And so what I'm going to show you here is some data, actually not in an Alzheimer's disease model, but we're actually going to use the visual system because it enabled us to actually move ahead with these pilot studies incredibly quickly. And this was some work that was headed by Kevin Gutenplan, who I mentioned earlier. It was published last year in Cell Reports. And I'm just going to show you two pieces of data from this, which I think is a nice little vignette to understand our reasoning behind these studies, but also show you some important functional outcomes. And so in this particular set of data, what I'm going to show you is just the viability of neurons. In this instance, we're using retinal ganglion cell neurons, and we're giving them an acute insult by axotomizing them behind the eye. And we're going to be looking at the viability of the neurons in the vertical axis here. And three little sort of case studies here that we're going to do is, in the first instance, these three are looking at wild-type mice. And we know that if we axotomize a central nervous system neuron through activation of these astrocytes, these neurons will actually die. And this is something that we've used in a whole host of different acute and chronic models over the years. But one of the things that Kevin was interested in was if we took otherwise healthy neurons, in this instance, wild-type mouse with undamaged neurons, and injected some media that we knew was toxic to neurons when we did our cell-based assays, we call this A1 astrocyte conditioned media, we actually saw that in this red bar here, these neurons were almost completely viable. And this gave us the first piece of information that maybe this toxin that is secreted from these astrocytes alone is not sufficient to kill these neurons. And in this white bar here, we actually just have a control setting where we've actually just injected otherwise healthy, physiologically normal astrocyte conditioned media that's full of trophic support nutrients that neurons would normally require. In this second set of experiments, what Kevin did is he took a mouse model that we generated a number of years ago that is deficient for the ability to produce these neurotoxic astrocytes. And we had already shown, as I'm showing again in this gray bar here, that if we cause these acute injuries or axotomy to these neurons, they actually will not die because there's no activation of these reactive astrocytes. So this here lets us know that the injury alone is insufficient. We also knew, this is just a repeat showing that the toxic media themselves, if we don't damage the neurons, is non-toxic and our control media is non-toxic. But what Kevin decided to do was take this mouse that has no reactive astrocytes, he damaged the neurons so that we would impart some susceptibility, and then he artificially injected this or exogenously applied this neurotoxic conditioned media. And when he did this, he was actually able to mediate the death of these neurons once more. So this was the first piece of evidence that we had that you required both some susceptibility on the neuronal side as well as some active component released by astrocytes in the non-neuronal cell component here to drive the death of these cells. But this actually wasn't even the most exciting part of the study. This was the bit that sort of proved our hypothesis. But the exciting bit of the study was actually looking at these neurons here. And these are the neurons that otherwise would have normally died, but we had maintained. And we were hoping that they would actually have some important physiological function that would give us the impetus to try and develop novel therapeutics to treat a whole host of different diseases. So we did this work in collaboration with Ben Stafford, who's based at the Salk, and also a lot of this data was analyzed by Drew Adler, who was an MSTP student here at NYU. And in these experiments, we were actually doing whole patch clamp recordings of these retinal ganglion cell neurons. In this instance, we're using a chronic model of neurodegeneration, so a mouse model of glaucoma. And what we saw when we did these electrophysiological recordings, at the end of the recordings, we dye-filled these neurons and then did some 3D reconstructions to look at what their morphology looked like. I'm just going to show you two things here. The first is if we look in an uninjected or a normal mouse eye, these retinal ganglion cells have dendritic arborizations that sit in a single layer in the retina. But if we induce glaucoma by elevating intraocular pressure, we can see that while these neurons stay alive, they're now making dendritic arborizations in abnormal regions throughout this part of the retina. And indeed, if we look in the Z projection, we can see that these cells normally have rather large arborizations, and in the Y projections, they have this single layer of dendritic arborizations. But if we induce this susceptibility but stop the cells from dying, we can see that they decrease their extensions as well as start arborizing in strange places. And importantly, we also did the electrophysiological recordings of these cells. And we can see that while in the control setting, these cells are extremely active, if we actually induce this disease model, but stop the neurodegeneration from occurring, where we expected there to be no electrophysiological function whatsoever, there was actually a maintenance of some electrophysiological function, not normal, but some function. And indeed, you'll appreciate there's far less of these events occurring here. But aside from a decrease in the so-called spike rate of these events, if we looked at each individual event on its own in isolation, the decay, the latency, etc., was all exactly the same for each of these events. And indeed, the retina's ability to actually measure gradations of light passing across the retina in different directions was not perturbed at all. So this gave us a nice piece of evidence that keeping these neurons alive by treating astrocyte-specific functions was actually beneficial in this particular model. And so we're interested in whether or not this would occur in other models of disease as well. So rather than using our visual system, which was very, very quick and occurred within a month, we could look a step further along the sort of chronic degeneration pathway and decided to look at something a little bit longer. And the model that we decided to use was actually a mouse model of amyotrophic lateral sclerosis, which we heard earlier when Moses was asking Helen a question. And the model that we chose was actually this mutation in the superoxide dismutase 1 gene. So this model is the SOD1 G93A high copy mutant. And what Kevin did here, and again, this was published last year in Nature Communications, is he looked for markers of these neurotoxic astrocytes, this so-called complement component C3. And he knew if he looked in the spinal cord of wild-type mice, there was none of these C3 positive astrocytes present. But as expected, when we looked in the SOD1 G93A high copy mutant mouse, there are a lot of these astrocytes present, presumably releasing neurotoxin. And so in a set of rather long crosses, Kevin actually produced a triple knockout mouse that didn't express any of the activating factors to produce these neurotoxic astrocytes, and then crossed that to the SOD1 ALS mouse. And when he did this, he saw a marked decrease in the number of these C3 positive cells in the spinal cord. And of course, the big question is, does this actually do anything? Is this beneficial for these mice at all? And indeed, if we look at the survival curve of these mice, so in the vertical axis here, we just have the percent survival. And we can see in the red line here, which is the straight SOD1 mice, as is the model, they start to die within 3 to 4 months' time. But if we actually knock out the activating factors for these reactive astrocytes, we can increase their lifespan by about 50%, which is one of the largest increases in life for this mouse model reported today. And not only that, we were interested, how good is this mouse model actually recapitulating the astrocyte reactivity we actually see in human patients? Or is this just an artifact of a mouse model and not important for our patient cohorts? And so what Kevin did is he took some human postmortem tissue from non-symptomatic or non-neurologic controls. He was staining for a marker for astrocytes, to glial marker GFAP. So we can see that there are astrocytes present, but we see in the non-symptomatic control patients, there are no neurotoxic astrocytes present. But if we look in patients that are harboring a mutation in the SOD1 gene, similar to our mouse model, we can see that these C3 positive cells are present. But indeed, if we look in other disease etiologies, we can see patients that harbor mutations in C9orf72, or those that are otherwise sporadic, we can still see that these C3 cells are positive. So that suggests that this neurotoxic subtype of reactive astrocytes doesn't depend specifically on an individual gene mutation. It's common across the mutations in these patient cohorts. And indeed, if you looked in the spinal cord, in the medulla, or in the motor cortex, all 3 regions associated with some form of degeneration in these patients, we can see an upregulation in the C3 positive astrocytes. And although this is only a small number of patients, if he looks at whole genome sequencing of several hundred patients, he can see that there is an increase in the amount of C3 expressed by ALS patients compared to non-neurologic controls. And again, this was not dependent on the known disease etiologies. So again, whether the patients harbored mutations in SOD1, C9orf72, or because of this other larger cohort, we could look at other genes associated with the disease like FUS, TDP43, or once again, if they had no known genetic etiology for disease, or these so-called sporadic patients. So the other thing I wanted to quickly talk about, now that we know that these cells are having a really important function, we were really interested in what other subtypes of these reactive astrocytes might actually exist. And this is something that we have thought about for a number of years, but haven't really had the capacity to investigate with any large endeavor. And the sort of support that we've had here at NYU, particularly from the Frangioni Foundation, has enabled us to do this. So what I'm going to show you is a couple of vignettes now of looking at heterogeneity of astrocyte responses, both within disease models and across disease models and in human patients as well. So this actually first brought up a lot of discussion in my lab, and everyone was excited to do this study, but we noticed very quickly that there was a problem when studying astrocytes using single-cell transcriptomics. And this problem really came down to a capture problem. When we looked at all the published data sets, they simply weren't capturing enough of these astrocytes to make these studies properly powered. And just to show you what this looks like, if we actually look at this very original paper describing DropSeq as a way of, or droplet-based RNA sequencing, even though this study sequenced close to 700,000 cells across a whole host of different brain regions, the capture rate for astrocytes was incredibly low, around 3%. But if we look in some particular brain regions, we know that the percentage of astrocytes is far, far higher, like in the cortex, where it may be 30 to 50% in some regions. This was then followed up by a number of studies, and I've only picked out 3 here using more commercially available 10x sequencing, looking in different brain regions such as the nucleus accumbens, the hippocampus, or the cortex itself, looking across different mouse models. In this instance, 2 studies using the 5x FAD mouse model of AD, as well as just treatment with morphine. We can see again the capture rate for these astrocytes is incredibly low, which doesn't seem like too much of a problem, but when we actually look at the number of cells that are captured per individual animal, we see this is also incredibly low. And if you then subcluster these astrocytes, look at subtypes of them, we may only end up with a small handful of cells in each cluster, which is really not sufficient power. So Philip Housel in my lab actually worked out a new method of increasing the capture rate for these astrocytes. We're now capturing close to 90% of astrocytes. We have an incredibly large data set, which represents actually the largest data set at single cell level resolution of astrocytes in the mouse, and enables us to have several thousand cells from each individual animal. And so what does that look like? Well, we actually did this by taking advantage, or Philip did this by taking advantage of an astrocyte reporter mouse. He's able to pre-sort for these cells and overload the 10x system to capture an incredibly large number of astrocytes, as we're showing here in red, with very small numbers of contaminating cells, which unfortunately for all the neuron and oligodendrocyte and microglia people in the audience, we then exclude and throw away. But when we get our 80,000 or so astrocytes, Philip can go in and see that if he looked at both male and female mice, either with or without an inflammatory insult, because we know a lot about this particular response, he could see that they express canonical astrocyte markers, and they also had very cluster-specific enrichment of differentially expressed genes. So we can now go in and interrogate each one of these individual substates of reactive astrocytes and determine if they have particular functions or they live in particular regions of the brain. And so I'm just going to focus in on one of these clusters here, this cluster here, which is an interferon responsive cluster. It's called cluster 8, or affectionately in the lab, we call these DUMAs, because why not? And what we know about these DUMAs is that they are largely only present in the inflamed brain. Here we see them here. They are largely identified by interferon response genes like STAT1, IGTP, and TAP1. And indeed, when we go and actually use in situ hybridization to see where they exist in the brain, they sit in very, very specific regions. And so here, Philip has done some in situ hybridization for IGTP to label these DUMAs. He's co-localized it with the astrocyte marker GLAST, or SLC1A3. And we can see that they exist only in layer 1 of the cortex, with their processes wrapped around these penetrating vessels. And just to show you that they're not actually endothelial cells, he's also co-labeled with the endothelial marker Claudin-5. And we can see the cell bodies have shrunk down onto the endothelial cells, but the IGTP itself is not expressed by those blood vessels. So this is just to highlight that we've generated this incredibly large single cell data set that we can use to generate differentially expressed gene profiles for each one of these different clusters. But Philip went one step further and actually used spatial transcriptomics to identify where each one of these clusters would actually sit in the brain. And I showed you this particular cluster here, this cluster 8 of or Duma cells, but I also like to pull out these particular clusters, cluster 3 and cluster 7, which have almost diametrically opposed expression locations in the thalamus and hypothalamus. And so we know now that the heterogeneity of these astrocyte responses is dependent both with respect to sex and time, and also their spatial location. And we're following this up now, looking at how they may aggregate differently around disease pathologies like amyloid plaques in AD. So the other thing that we did, because 2020 was a rough time, and so we had a lot of time to do coding, is we made a very easily searchable website. So you can go in and search for your favorite gene at the single cell level, and also using the spatial transcriptomic data that we generated. So you can see astrocyte upregulation of genes across the entire brain or in specific subclusters of these cells. And this is a different subcluster of reactive astrocytes that exists only in this white matter tract that underlies the hippocampus. And we were also able to integrate these data with other disease models. And so this was done by Jessica Sadik in the lab, who integrated with these 5X FAD mouse models from Naomi Habib in Mikhail Schwartz's lab, and also from Jinju in Marco Colonna's lab at WashU. And while these data sets were extremely underpowered themselves and couldn't identify the clusters that we discussed, we found that if we integrated them with our large data sets, we could see that these interferon responsive Duma cells, actually with a number of markers, were present in the early stages of AD pathogenesis at 7 months, but not in the later stages at 15 months, suggesting these are really important in the early stages of etiology of this disease. And because we're very disease agnostic, we also looked at a demyelinating mouse model, the EAE model of multiple sclerosis, and found that these astrocytes were also present there, and they were present in this acute stab wound injury in the mouse brain as well. In all of these data sets, these astrocytes had not been described previously. And then in my very last slide, I just wanted to show you some work that we did with single nuclear sequencing, thanks to the tissue bank that's available at the ADRC just downstairs here in the science building. And Jessica Sadik actually developed this very large data set of human astrocytes and also oligodendrocytes. She could then integrate it with data sets that have already been published that were extremely underpowered. And then by integrating these data sets together, she can see that there are actually some really important physiologically distinct subtypes of these reactive astrocytes that have been under sampled and not discovered in previous data sets. The last thing that we can then do, because of the benefit of Big Purple here at NYU, is we can actually now integrate our mouse data sets with our human data sets, which requires a large amount of computational power. And we could determine which substates in our human data sets from patients are actually recapitulated in our animal models of disease. And this is just to show you two examples of this top cluster here is actually showing us how these Dumas are present in both human patients with AD and in our mouse models. And also these neurotoxic astrocytes are present in human diseases and also in some of our animal models as well, which gives us hope for actually using these models to study these cell subtype functions going forward. So that's all I really had to say. I just wanted to quickly give you an overview that we know that these reactive astrocytes have both transcriptomic and functional heterogeneity, and that depends on spatial location. And this will be published in Nature Neuroscience in the coming weeks. We also know that some of these mouse models of diseases do a very good job of recapitulating the human condition. And I showed you some data from glaucoma and ALS mouse models. And we also know that these neurons must be susceptible to be killed by these astrocytes that are releasing a neurotoxin, and that these neurons, once saved, maintain some electrophysiological function that we think is really important from a therapeutic avenue. And I also only briefly mentioned, but very importantly mentioned, that while we did a lot of this work in rodents, we know that this particular mechanism is evolutionarily conserved from rodent cells through to human cells as well. With that, I am done. I thanked all of the people going through and highlighted them when they did the work. I would also like to acknowledge all of our funders, particularly the Blas Frangioni Foundation and the ADRC here at NYU, as well as all the donors and families who have donated tissue for us to do these studies. So thank you very, very much.
So thank you, Shane, for a wonderful talk. We're now open for a couple of questions.
Mitchell, I allowed you to talk if you want to.
Okay, yes, thank you.
Hey, Dr. Liddelow. First, thank you for the talk. Definitely really, really nice. I have kind of two questions related. So basically, and you show it here, there is this sort of feedback loop in between the astrocytes and the microglia. So microglia is releasing certain cytokines. It's turning the astrocytes into these angry neurotoxic ones. But then the astrocytes, they release molecules such as the complement 3 to like, at the same time, they signal to the microglia. So my first question is, does this happen with the neurons as well? I mean, I know neurons have certain receptors for the astrocyte molecules, but do the neurons also feedback the astrocytes? Do they send certain signals?
Yeah, that's a great question. I'm sure they do, Mitchell, but we actually haven't focused on the neurons at all, which I'm very embarrassed to admit. We know that neurons do secrete alarmins or stress molecules under response of these sorts of injuries and insults. Actually, neurons secrete complement component C1q under stress. So there's a whole host of molecules that they do release. We haven't looked at what the effect of those is on astrocytes or on microglia per se, but it's something that we should look at down the line. It's a really, really good question.
Thank you. And just a quick second question is, these already neurotoxic astrocytes, thinking on, I don't know, a therapy or something, could they go back to a quote unquote normal state, or are they just like lost?
Yeah, that's also a really good question, and I skipped all of that. So we do know that the astrocytes can revert back to a physiological state. We've done this a number of times, both in our culture systems, but also in some of our acute insult and injury models. This can be done as simply as using things like FGF or using some anti-immunotherapy treatments as well, but it's not as particularly well defined as one would think. We do have a lot more hope in actually stopping them from forming. So there's a glucagon-like 1 peptide receptor agonist that we actually trialed in a paper with Ted and Valina Dawson in a mouse model of Parkinson's disease, and showed that it was very effective in inhibiting this microglia astrocyte axis to stop the neurodegeneration. And that's now just entered phase 2 clinical trials for Parkinson's disease and phase 1 trials for diabetes. We have another paper that will be out very shortly where we've identified the neurotoxin, and we actually have the ability now to stop just the enzyme in these astrocytes that produces the toxin and stops its release. And we think that that is a much more beneficial target moving forward, as there are no current therapeutics for that target. So it's a very, very novel way for us to approach this moving forward.
Thank you so much.
I think a question by Dr. Chen. Oh.
Can you hear me now, Shane?
Oh, got you, Dick. Yes, I can hear you.
I wasn't quick enough to catch the part about sex differences.
Oh.
Which part of the brain was that found in? Do you think it's going to generalize across all regions of the brain? And what's the relationship between that and what we know about disease?
Yeah, that's a really good question, Dick. So we did all of those initial studies in the cortex, and we were really excited because we thought there would be very big sex differences, and actually they were very, very subtle and almost non-existent in the cortex. We have now gone back and started an incredibly far more thorough investigation at the single cell level across sexes in all brain regions, and we're actually pulling out some interesting differences there. I will say that one of the biggest differences is not the heterogeneity of the reactive states that occurs, but the timing of that response. So there's differences in the speed of both male and female astrocytes to respond, which may actually be a result of the known sex differences in the microglial responses have been worked on by people like Stacy Bilbo at Duke University. So there's a lot more for us to do, which means a lot more sequencing, which means us giving a lot more money to Adriana over at the GTC, which is great. And it'll also mean me walking downstairs and getting a lot more tissue samples from Tom and Arlene. But it seems that the astrocyte responses on their own are not super sex specific, but it's the microglia feeding into it, if that makes sense.
Thanks.
So thank you for a wonderful talk, Shane, and a good discussion. And again, there'll be opportunity for more questions in the question and answer session.